We're right in the middle of fermentation, and I think as you were departing last time, I asked you to do an assignment. I probably should have just collected those real quick, but go ahead and pull that piece of paper out of your notebook that has the answer to the question you were given, and pass those up real quickly so we can get going. Make sure your name's on it so I can give you credit for having completed that assignment. If it's got your name on it, that's fine. Did you tell me how many carbons are in it? All right, beautiful. Pass them up, please. You can just sit on top. Oh, you got one too. Pass faster. If I get there and it's not passed, it's not passed. It's already passed. Did you guys get yours up? Dude, that's way slow. What is that? There's no name on it. That's all right. You don't get credit because there's no name on it. Okay. I'm just messing with you. I'll try to remember that it was yours. So the question I asked you, or the assignment I gave you, was to go look at Wikipedia and figure out how many carbons coenzyme A has. I actually got an email last night at 11 o'clock, maybe 11:30, asking to confirm. And so what'd you come up with? 21 for coenzyme A, right? I should have asked you too how many nitrogens there are. There's a good number of nitrogens, 7, 8 nitrogens in there, and there's some sulfur in there. Did you figure out where the acetyl group attaches to that when it becomes acetyl-CoA? Did you go look at that? Let me see if somebody put a picture on here. So, yeah, this is the straight coenzyme A. There's the molecule right there. And the acetyl group, 2 carbons attached right to the end there. Actually, wait a minute, that's got the acetyl group on it already. Normally, if it's just coenzyme A, that sulfur that's in the last position is going to have a hydrogen on it. And this one's already got 2 carbons attached to it. So this is actually acetyl-CoA. And so if coenzyme A has 21 carbons, how many carbons does acetyl-CoA have? 23 carbons. Why? Because an acetyl group is a 2-carbon molecule, and you add those 2 carbons to that sulfur group that's on the end there. Beautiful. My whole purpose of having you look at that was to get you a little bit familiar, to hopefully create some curiosity, to realize it's not too hard to dig in. When you hear a name that you don't know, it's not too difficult. It takes maybe 3 minutes to go and look for what that molecule looks like. So in this class, you're going to get lots of new vocabulary. Some of it's going to go here above your head, and you're not going to know. But if you just jot down a note and write the name that you heard, you can very quickly go look at it later and figure out what that looks like and how big it is. And most students, when they come out of anatomy and physiology, they say acetyl-CoA is a 2-carbon molecule. That's what they got from that process. And they don't realize how big the carrier is, how big the coenzyme A is. So I really wanted you all to know it's actually a relatively large molecule. There's something else peculiar about it. Did you notice most of them draw it with over on the right-hand side, but there's a double ring structure up there on the right-hand side. Did you notice that? What did that remind you of? Not a protein. It looks a lot like a nucleic acid. Actually, it's adenine, one of the bases, attached to a ribose that actually has a phosphate group attached to the number 3 carbon. So if you were to go look, how many of you have ever gone to look at the structure of NAD or NADH or FADH2 or FAD or NADPH? Have you looked at any of those before? Do you have any idea what they look like other than an alphabet soup? Yep. You got some ideas? Yeah, there's similarities between all 4 of those molecules. After class, sometime you should go look at them. They all, remember we said even those guys, NADH and NAD, FAD, FADH2, NADP, NADPH, they all can function as coenzymes. Go look at those structures and figure out what the similarity is to this guy and how they might function as coenzymes. Just go look them up. I'm not going to require that you pull them out on a piece of paper. I'm hoping I'm piquing your curiosity so you can realize, hey, I can go look things up that I hear about and understand them more fully, and it will make things make more sense. It will help me understand the material that we're talking about. I really encourage you to go look at those 3 additional molecules that we've called electron carriers so you can get an idea of what they look like. It'll also help you remember their full names and not just the 3-letter or 4-letter abbreviations for those particular chemicals. All right, so we were here talking about fermentation last time. And fermentation we can use not for the things you're thinking of. Well, maybe some of them. We use fermentation all the time for a lot of food products. And when I lived in China, I could do one of those crazy food shows where you go eat all the bizarrest things, and because I've eaten most of them. I had camel hoof, I've had dog, I talked about that in lab yesterday. I've had silkworm worms. Those are okay if you get over the fuzz. And my favorite of all the silkworm life cycles is the silkworm butterfly. If you deep fry those and add the right seasoning and close your eyes and eat them, they taste a lot like Lay's potato chips. And I'm talking to you about this because Chinese ferment a lot of interesting foods. And there's one food that, how many of you had that? If you've for sure seen the commercial, they're usually little cube chunks of gum. And the commercial says you bite into it and you get a squirt of freshness in your mouth or something like that. You've had that kind of gum before? That's pretty delicious, right? Well, there's another form of silkworms. It's the larval form. And they're big and black and round, about that big. And any Chinese person will tell you they have more protein than 3 hard-boiled eggs. And I don't care how much protein they have, they taste terrible. When you bite into them, it's just like that chewing gum. They boil the cocoons, the larvae, and then if you ever get invited to a banquet in China, they're going to invite you to enjoy one. And if you don't, it's an insult. So I had to go to lots of banquets because I was the director of a couple different companies, and so the government was always inviting me to banquets. And every time they'd say, have you had one of these? And after a while I could say, oh yeah, I've had hundreds of them, I love them. Then they won't just put it on your plate, and they'll let you decide if you want to take it. But what you do is you put it in your mouth, and it's pretty big. You can't eat it all at once, but if you try and bite it, it's just like that chewing gum. You bite into it, and nice juicy stuff squirts out into your mouth. Only the flavor is exactly the opposite of that chewing gum. You don't want to bite it anymore as soon as it squirts out. But there's lots and lots and lots of protein in there, so it's very good and healthy for you, according to Chinese tradition. So I'm telling you this because they've got all kinds of fermented foods. That one's not fermented, but they ferment just about everything. Cabbage, anything you can think of, they ferment. They've got some eggs that are fermented that supposedly will last for 1,000 years, and they've got all kinds of other things that they ferment. And so there's lots of things. But we eat some things that we think are a little bit less bizarre than some of those things that I just described. So none of us do this stuff right here, but you might have friends that consume beer or alcohol, and that's a fermentation process that does that. And most of the time it's Saccharomyces that does that. But if you've noticed any of the advertisements that show up on TV and other places, every brewer is trying to get their own priority, whatever it is, trying to get their own flavor that they can call their own. Proprietary, there it goes, my tongue finally agreed to cooperate. Proprietary flavor that they can call their own. So they put yeast in there. And in fact, if you're interested, you're not, but if you were, you could go look, and there's 1,000 different strains of yeast that each do fermentation a little bit differently and give a little bit flavor to that concoction that you're making. And then some people actually add bacteria of different strains and varieties, because when they do fermentation, they make a few other products, and those other products give the final product a unique flavor that someone might say, ooh, that's interesting, I want to try more of that. And so they use a lot of different strains of bacteria to be able to produce those liquid beverages. And as we talked about, different organisms do fermentation in different ways. And you've got just 6 of them listed there, but if you imagine there's 100,000 organisms that can do fermentation, each one of them is going to do it slightly differently. Get back on something that we can eat at UMHB. How many of you like Swiss cheese? I love Swiss cheese. It's probably my favorite right after pepper jack. If they made a pepper jack Swiss cheese, then that would probably be my favorite for sure. But why do you like Swiss cheese? I saw some of you raise, probably most of you raised your hands at that point. Why do you like Swiss cheese? What's different about it?
It's a little more bitter, or not exactly. I don't know, it has a different flavor
How would you guys describe that flavor? Like cheese? Tastes like cheese should taste.
No, I said I just like cheese.
Oh, you just like cheese all the way. Okay. No cheese. My little brother hates, my dad worked for a dairy, and he hates cottage cheese. I think it's the texture, but I like cottage cheese. Put some cinnamon sugar on it, but that's another story. So why do you don't like it? Try it with cinnamon sugar on it. It's delicious. Not the Swiss cheese, but why do you like Swiss cheese? I've never tried it with Swiss cheese. I may do it tonight.
I don't like Swiss cheese because it's too sour.
It's too sour. It's that sour flavor. Some people like it, and you said bitter. A lot of people describe it as nutty or fruity flavor, and that's what's unique about it. The bacteria that makes that flavor is called Propionibacterium, and there's a number of different species that are used. So what do you think makes those holes in the Swiss cheese? Gas bubbles is more accurate. The bacteria are put down into the curd, into the material, the milk material, and the bacteria there are in an anaerobic environment, and they start to do fermentation. As they do fermentation, it's listed for you there, they start to produce gas, and the one that they make is this carbon dioxide. So they're growing there. They literally make a nice little colony, and the colony starts doing fermentation, and as it does fermentation, it makes more and more gas, and it makes a bigger bubble, and pretty soon they add a chemical to make that liquid environment gel, and the bubble is trapped down inside. So next time you have a piece of Swiss cheese, you're probably just going to have a slice. You can still see it. Look around the edge of what was the bubble, those holes in there. Have you ever noticed it's a little bit different color? Lots and lots of bacteria down in around this circle, along the circle edge. They're there sitting, fermenting. They've got to be on the edge to get the nutrients. They're not outer space creatures, so they can't float inside of the bubble. But if you look at the edge of the bubble, they're growing. And they make this a couple of different acids. One is acetic acid. That one your taste buds are familiar with because that's the acid that's in vinegar. The one that your taste buds probably don't experience many other places that the Propionibacteria make is propionic acid, and that's how they get their name. These organisms that make propionic acid are called the Propionibacterium. And so they put those in there intentionally to make that, and that's what gives it the nutty, fruity, kind of tangy flavor. And so one thing that I told you about Chinese foods, I need to tell you a little bit about Chinese language. Chinese language is really difficult to learn. People say that. I don't know. For me, it was the easiest because I got hungry. I wanted some cheese, and I had to figure out where I can buy it, and they don't eat cheese, so I had to ask more people, and I got to talk to lots of people, got more hungry, and so I had to ask more questions. I had to learn how to say those in Chinese, and so I learned to talk out of necessity in Mandarin. And so, but the language is really, really intuitive, really makes sense, because every word has a literal meaning. And so for acid, the word for acid, no matter what kind of acid is, it's called swan. Swan means acid. So you'd have hydrochloric swan or acetic swan or whatever it is, and they'll put another modifier sound at the beginning of it to tell you which kind of acid it is. But the language is so logical. It makes so much sense because there's that one word for acid, but there's a word for sour too, and it's the same word, because they know that acids taste sour. So they use exactly the same word, whether they're talking about some kind of acid or whether they're describing a sour flavor. And so a lot of things in the language are actually easy once you figure out, oh, an acid does taste sour when you taste an acid. And I looked the other day, it was actually a few months ago, but I looked at our potato chips bags in the grocery store because we're starting to do bilingual on the things that are in the grocery store with English and Spanish. And on the sour cream potato chips, on the back it says crema asada. I think I pronounced it close to right. Acid cream on the back is literally what it says. I don't know if that sounds beautiful in Spanish or not. In English, we don't really think about what sour cream is saying, right? If you thought about it, you'd say, ooh, I don't want to eat something that's sour because sour milk is not very good. Why is sour cream any better? But that's what the potato chip wrappers say. And so I just want you to understand acids do taste sour, and that's what gives flavors to a lot of these products that we're talking about, the different acids that are produced. We can do a lot of different tests. In fact, you're going to look at some tests to see does the organism make acid or not. You won't be able to tell which one, but you'll use that test down the road. If I give it this kind of carbohydrate, does it make acid or not? How does it use that carbohydrate? And there's lots of different ways. If you feed E. coli only pyruvate, they'll make a number of different acids. You can see those there: acetic acid, lactic acid, succinic acid, ethanol, and then they make a couple of gases. So you're starting to get a picture. The products of fermentation always include acids, many times alcohols of different types, and if you look through that list you'll see a lot of different alcohols, and or gases. Not all the different pathways make all of those. Some of them do, some of them don't. It just depends on the enzymes that they have to do the fermentation process. So that if you look at Saccharomyces, when it does fermentation, and that's the reason people put it in their beer and their wine, it makes alcohol when it does fermentation. It doesn't make the other acids nearly as much, and then it releases CO2. The lactic acid bacteria, those guys make lactic acid. I need to tell you about the lactic acid bacteria just a little bit. This relates back to lab. So about 20 years ago now, people were collecting lots of different organisms, lots of different strains, and everything that was cocci and made chains of cocci were called the streptococci. And the group got humongous. So many organisms that it was hard to keep track of which one it was, and it probably was hard to think of new species names that hadn't already been used by somebody else because the group was so big. And so the scientists that were working on nomenclature and naming organisms said, this group's way too big, can't keep track of it all, can't make names that are logical anymore. We need to break up the streptococci into smaller genus groups, and the logical way to do it is to break them up based on where they're typically found. So the streptococci that are found in milk and dairy products became the lactococci. Okay? The streptococci that are found normally in the gut became the enterococci. Okay? Entero means inside. And so you guys actually played with an enterococcus the other day. Enterococcus faecalis, the species name tells you where you're going to find it inside of fecal material, right? Enterococcus faecalis. And so it was originally called Streptococcus faecalis. And then when they split them up, they changed the genus name to Enterococcus faecalis. And then, so that was milk, and that was the gut. And then they said all the other ones that are out there, wherever we find them, we'll still call those streptococci. So the Streptococcus pyogenes that causes strep throat, and all those others that exist in other places are generally still called streptococci. So they broke that big group up into 3 different genuses based on locations where those organisms prefer to be growing, and that makes some sense. So that when you think about that in the laboratory, you can think about what you're playing with and what you're using there. But basically you could go, if you had a mass spec available to you, you could take the fermentation products, you could pump those fermentation products into a mass spec or a GC, and you could figure out which of those chemicals are being produced, and you can use that to ID a bacterium. We'll talk about bacterial ID in the next chapter, or the next couple chapters. So you know that microbes could use a variety of compounds in their environment. They're going to take advantage of whatever. I think I've told you guys that glucose is most bacteria's preferred Snickers bar. You know the Snickers commercial where there's the guy in the back seat and he's whining like an old lady, and they say, come on, eat a Snickers bar, and he takes one bite and he's back to his normal self again. And so if you put bacteria, almost any bacteria, in a tube and you feed them glucose, you feed them lactose, you feed them mannose, you feed them 100 other sugars, they're not even going to look at the other sugars until all the glucose is gone. They're going to eat the glucose first, and then they'll start eating the other sugars. And we'll talk about how that happens down the road. But sometimes out in the nature, though, nobody feeds them glucose out in the environment, so they have to make their own. They have to hydrolyze macromolecules, and if you look back to Chapter 2, you learned about a bunch of macromolecules that exist in the environment that can be hydrolyzed by these organisms or broken down into monomers, and then the monomers can be taken down into the cell for energy. So to do that, they have to secrete those enzymes outside of the cell, and then they have to have some transport system to bring them back into the cell. And then those metabolites are going to be then further broken down to make the various precursor metabolites. We've already talked about another way that they might do that if there aren't any nutrients in the environment. Remember that process? What happens to bacteria when they get hungry, meaning there's no nutrients in the environment? Some of them might make spores if they can do that. They start using all of those molecules they put outside when they were growing fast, like the LPS and like the peptidoglycan layer, the glycan chain. They can break those down too. They've got enzymes to break those down and bring those carbohydrates back into the cell if they need to. So they can use polypeptides and disaccharides. They can actually use starch, and the enzyme that breaks down starch is called an amylase, and there's lots of different amylases. They can use cellulose, and there's lots of different cellulases that are out there that can break these polymers into monomers so that they can be brought into the cell. And then the enzyme that would break down the disaccharides is going to be a disaccharidase. And those guys would break down these disaccharides. Lipids can be used as an energy source too, and to do that you need to break down the fatty acid tails. You need some lipases that can break down the fatty acid tails, and then the glycerol can be converted to dihydroxyacetone phosphate, which is the product in glycolysis just after the branch step. So it can feed in just after the branch step. It can go on down through glycolysis. You actually get some ATP out of that dihydroxyacetone phosphate right out of glycolysis. And then it can feed, the carbons can feed into the TCA cycle and turn the TCA cycle for a turn. And then the fatty acids, when they're hydrolyzed, they're broken down by a process called beta oxidation, and then they can feed into the TCA cycle and turn the TCA cycle that way as well. So all of those macromolecules that we're talking about building through anabolic metabolism can be broken down to release the energy that's stored down inside of those macromolecules. Proteins are the same thing. Proteases can hydrolyze those into amino acids, and then the amino groups can be deaminated, and then they too can go back into glycolysis and be used in glycolysis for energy once again. Carbon skeletons can be converted to precursor metabolites. So this is a beautiful schematic picture. I think I told you last time that you need to be familiar with table 6.2. Probably familiar is not the right word. Table 6.2 and figure 628. Did I tell you guys that? Have I told you that? Some of you are acting like I didn't. In fact, I'm not seeing any confirmation that I did. So for the final exam, you get to tell me some very specific stuff. And that table 6.2 that I think we looked at last time has all the precursor metabolites that are produced out of the central metabolic pathways. You want to know that for the final exam. It makes me cry every time one's turned in blank on the final exam, because if it shows up on your final exam this year, it'll be worth a significant chunk, almost a full letter grade. And if it shows up blank, that means I can't give you that full letter grade on the final exam. So you really want to invest a little bit of effort studying those precursor metabolites that are listed in table 6.2, and they're also listed in 628 specifically. They're listed in this figure that we're seeing here, but they don't give you the names for any of those. Each one of those little ovals that's listed here is a precursor metabolite that comes out of this pathways. So that helps you start to get the idea that, hey, they all come out at different places in the pathways. But what this figure is trying to show you is that those macromolecules that we just talked about can be broken down. For example, here's the polysaccharides. That includes things like cellulose. It includes things like starch. Those can be broken down by the appropriate enzymes, and then once they're broken down into monomers, the monomers can feed back in one place or another into glycolysis. And once they feed into glycolysis, they can flow through glycolysis and drive glycolysis in the TCA cycle, and all that energy can be recovered back out of them. Same thing for disaccharides like lactose. That's one that you're familiar with. And maltose, sucrose is the one you use for table sugar. They too can be broken down. The enzymes that break them down are called disaccharadases, and the name is right here. And again, they're going to break them down into monomers. They're going to feed into glycolysis, and it's going to drive glycolysis a couple of times. And you get all of that ATP from the reducing power that is released as those are being broken down as well. Lipids come over this side. You remember lipids contain glycerol as one component. How would you describe glycerol? What does it look like? How many carbons does it have, since we're thinking numbers of carbons now? How about if I say less than 5? How many does it have? 3. Beautiful. Glycerol is a 3-carbon molecule, and those lipids, whether phospholipids or simple lipids, are going to be broken down. We're going to have glycerol for the backbone, and the fatty acids are added on to that. So when lipases get a hold of lipids, they break the bonds that attach, those ester bonds that attach the fatty acids, and the fatty acids are released. They can be broken down by beta oxidation. They come in, the 2 carbons come in here, so they're broken down 2 carbon by 2 carbon by 2 carbon. They feed in here and are attached to this acetyl-CoA, and that drives the TCA cycle again as well. Okay, so that figure is just showing you how macromolecules can actually be pulled in. Proteins do the same thing. Proteases break them down, and there's lots of different places that those amino acids feed back in to the TCA cycle and the transition step and the glycolysis pathway or glycolytic pathway. So keep that in mind, that out in nature, organisms don't get fed those pure chemicals. They've got to deal with macromolecules that those usually photoheterotrophs are producing out in the environment. There are lots of prokaryotes that have unique abilities, because there's a lot that have a good bit of diversity. One group I'm pretty sure you haven't thought about before are these guys, chemolithotrophs. You learned the name and what it means the other day. Do you remember what chemolithotroph means? You've got to think about 2 things. Where do they get their energy from? Inorganic, right? That's what the litho part tells you in that name, is that they get their energy from inorganic molecules. And these guys typically are going to be heterotrophs, even though it's not listed on this slide. They're going to be chemolithoheterotrophs, meaning they're going to get carbon out of other molecules, unless they're chemolithoautotrophs, and then what would they do? Produce their own, what they eat, their own carbon. So if they're chemolithoautotrophs, they're fixing CO2. That's what an autotroph does. The other part, the types of autotrophs that you're familiar with typically use sunlight for energy. These guys, some of them can use energy that can be reduced from inorganic chemicals.
Something like that live?
Where?
Where would something like that live?
We're going to look in just a second, and I think I'll answer that question, because they're going to live in all kinds of different places. They're going to live wherever the chemical is abundant that they can get energy about. You probably forgot, because it's got the word margarita in it, but you remember that Thiomargarita nambiensis? It lives in Nambia. It lives out in some marshy areas, and there's a high sulfur content in those marshy areas, and it's one of these guys, these chemolithotrophs. It gets its energy from those inorganic chemicals. Remember, it's got 2 chemicals that if you put them in a test tube would actually spontaneously ignite in a test tube, but it sequesters those down inside of the cell and lets them come very slowly back together. And it can harvest, it's got enzymes to harvest the energy out of those chemicals as they react with each other in a controlled fashion. So they live wherever the chemical that they've adapted to use or were designed to use exists in abundance, that they can get enough energy. And well, let's look at this list, and that'll make things make a little bit more sense. Some of these guys take hydrogen gas. And I think I mentioned to you the chemistry club is going to do demos in the dark sometime this school year. It's pretty cool. You got to go if you haven't ever seen the demos in the dark. I like to go just because they blow stuff up and they make stuff glow. It's pretty cool. And if you got kids, you got to take your kids, because it'll get them interested in science. So, but one of the things they do is fill a bunch of big balloons with different gases, and one of the gases is hydrogen gas. And then they ignite each one of the different balloons. You can see that they burn different colors. You can actually hear that when they explode, they make different sounds. And you can get some ideas about the different levels of energy in the different gases that they're igniting. Well, hydrogen is a gas. We talked about the Hindenburg before. Remember that dirigible that they filled it with hydrogen so it would lift off the ground because hydrogen's light, and then it ignited and caught on fire. It burned, and everybody crashed in a burning ball to the ground. Well, some bacteria get their energy from hydrogen gas, and And the reason I mentioned demos in the dark is because most students can't imagine how a gas could have that much energy. But when you see that balloon filled with hydrogen gas, and then they just put a little spark by it, it's kind of a little spark, it ignites very quickly and all the energy is released very quickly. Well, bacteria can take that same hydrogen gas. They're probably not living down inside of that balloon that the chemistry club is using, but they find hydrogen gas out in the environment. And when the hydrogen gas is in the environment, they can do the same thing. They can do a redox reaction on the hydrogen gas and squeak out some of the energy that's in that hydrogen gas and capture it. So to go back and answer your question, where would these guys live? You already know about it because we just talked about it less than 10 minutes ago. Where do you think organisms that need hydrogen gas could live? Don't say those balloons that chemistry club uses. They could. They'd love it there, assuming they could get some carbon and all the other minerals. Excuse me? Yeah, that's right. They need hydrogen gas as an energy source. There's a little bit of free hydrogen in the air, but let's go back here to this point where we were discussing something earlier. Check this out. What's the product of fermentation here? And here. And here. So usually they live in combination with fermenters that ferment and produce hydrogen gas as a waste product. And then whenever that organism is growing, it's making that waste product and it's releasing the chemical that they need to be able to grow and survive. So that's those guys. And so hydrogen is a product of many different, hydrogen gas is a product of many chemical reactions, including fermentation. And then there are these guys that are called the sulfur bacteria. And their energy source is this hydrogen sulfide. What do you guys know that have had a chemistry class? What do you know about hydrogen sulfide? If you get a reasonable concentration of hydrogen sulfide in a solution, what do you think happens to the pH? Drops. It gets very near to 1, a pH of 1 if you get the concentration high enough. And so that tells you something about these guys automatically, that they're, many of them acidophiles. They like to live in a low pH concentration, and they do that because they need the hydrogen sulfide for their reactions. Look what they do. They take the hydrogen sulfide, they react it with oxygen, and they make water and they release sulfur. So next time you drive across 6th Street, drive slowly instead of speeding up when you get past McDonald's. And open your window and take in a few deep breaths and smell what you smell. Many times, it's not always this way, but many times when you drive past the wastewater treatment plant, you smell sulfur in the air. And it's because in the tanks where they're dealing with all the waste that comes through, they have a lot of these sulfur bacteria that actually release elemental sulfur, and it goes out into the air, and you can smell it, and you can recognize that. Many times along with those bacteria, there'll be other bacteria that can take that elemental sulfur, and they can then oxidize it a little bit further and get a little bit more energy out of it and make hydrogen sulfate. And again, those guys are going to be living down in a low pH. So again, these guys typically live in a symbiotic relationship with other organisms. One does one of the reactions, the other will do the other reaction, and they live together, providing the energy sources that they need. Then there are others that can just take iron, and they can take reduced iron. Remember, anything that's reduced has reducing power. They can take reduced iron, and all these things that we're talking about are reduced and they're being oxidized. You can look at those formulas and realize that. They can take that reduced iron, electrons are then moving, and the iron gets oxidized, and some energy is released out in the process. And these guys tend to live in a lot of different environments where there's a lot of iron in their environment. These guys, the nitrifying bacteria, are also interesting, and these guys, we actually give them beautiful environments every year about March, especially here in Central Texas and other places where there's a high agrarian society, where there's lots of agriculture. Because every year in about March, we take huge tank loads of this chemical, ammonium, and we pump it into the ground. Because ammonium is a chemical that can be captured by these bacteria, and then it can be incorporated into amino acids by these bacteria, and then the bacteria can feed that nitrogen to the plants, and the plants need the nitrogen so that they can make all the proteins that we need to eat when we eat those plants. So we pump tons of it into the ground, literal tons, probably teratons of nitrogen in the form of ammonium into the ground every year to give the plants the nitrogen that they need, and that's done separately by some nitrogen-fixing bacteria. Some of them will do this reaction, take ammonia and oxidize it, and then you get nitrate, and then you can take the nitrate and oxidize it to nitrite. I think I got those backward. And so usually those guys also live in a mixed environment. So we pump a bunch of ammonia into the soil, and it's usually good as long as it doesn't rain real heavily for 2, 3, 5 days or 7 days. But if it rains real heavily in that short amount of time, the ammonia gets sucked up into the water and it runs out into rivers, streams, lakes, and ponds, and you've got these bacteria that are living there. And look at these chemical reactions. What are these reactions doing? Besides, we say they're oxidizing those nitrogen-containing compounds, but look what they're actually doing. They're all consuming oxygen. And so when the ammonia gets absorbed in the water and runs into the streams, and the bacteria do those same chemical reactions, they're using up the free oxygen in the water. The water becomes, my brain's really going slow today, not sure why, becomes anoxic, low in oxygen. Okay? And so what lives in the water besides these bacteria? Fish. Fish need the oxygen, so the water becomes anoxic, low in oxygen. The fish don't have enough oxygen. The fish start dying, and then other bad things happen in those lakes and streams. And so that's something to consider. Right now we're just worried about the bacteria and giving them a healthy life, so what we do is actually a good thing. It gives us a good life too. It gives us food to eat in abundance. Yeah?
I mean, instead of spraying ammonia on the fields, could they just spray bacteria?
They could, but the bacteria are already there, and the bacteria need something to eat, and the ammonia is what the bacteria eat.
But don't
So,
we have a constant supply of nitrogen in the air, which is what they're
yeah, and
anyway, right?
if we didn't put the ammonia in the soil, the bacteria would pull some nitrogen out of the atmosphere, but they can't do it as efficiently as if we put a whole bunch right in their environment. So they do it much more efficiently. They grow much faster if we pump it down in there, and then they can feed the plants so the plants can grow faster. The process works fine. Obviously, it's done it for however long the earth has been in existence till we decided this will make the plants grow a little bit more quickly. It also, when we put extra in there, it boosts up the protein content in those products that are being made, and so rationally you get a higher product out of it because of the protein concentration is higher in those materials. And most of the time we do it for corn, and the corn is going to be used to feed cattle and other organisms like that. We want to have as high a protein concentration as we can.
Well, that's why they do the crop rotation too.
Yeah, sure. And so soybeans do they'll switch because soybeans add nitrogen back to the soil. And there's lots of ways to do farming, but some of them get faster crop and more crop faster. So it's all economics, and you can rotate, and if you can. Usually when you rotate, one crop gives you less product, less income than the other, but some farmers still do that because they know it's good for the soil, but others thinking, okay, I need to make money, and if I raise corn this year, even though I raised corn last year, I'm going to get more. Right now there's a great market for corn because we're using a ton of it to make gas, to make that 10% or 80% ethanol that's in our fuel, and so they're getting really high premiums on corn, and if they grow corn every year, the soil gets depleted of nitrogen, so we have to put it back into the soil. And if they could, and in my area, some of the farmers will rotate soybeans, corn, soybeans, corn, soybeans, corn. But they've got to, if the market for soybeans isn't very good, then they might lose money that year when they're growing soybeans. All right. So, what do you guys know about photosynthesis? Plants do it. Some do. All do. All do. Okay, who else does photosynthesis? Some bacteria can do photosynthesis. Good. Algae. Yeah. Okay. What is photosynthesis? Photosynthesis derives energy from photons of light. Is that all it does?
Make sugars.
Builds macromolecules. What's it build them out of? It's inorganic till they grab it. Yeah, what kind of carbon? Carbon dioxide. Yeah, we don't usually think of carbon dioxide as an organic molecule. Technically it could be because it's got a carbon in it, but it's kind of an inorganic molecule too. It's kind of that crossover in between. So they do that. Do they do anything else that's important to us? They produce oxygen. Good. So photosynthesis, harvests energy from light, fix CO2, and produces oxygen. I see you doing it. It's good. We all have to do it. That's all right. So is that the definition? How many of you learned about photosynthesis in 8th grade biology or 7th grade biology or maybe even 5th grade biology these days? A lot of you did. Did you learn somewhere along the way in high school you had a class that talked about photosynthesis? You probably talked about the carbon cycle, how we do respiration. We break down those organic molecules, and as we do respiration, we release CO2, and I talked about the first day of this chapter as well. We release that CO2, those photosynthetic organisms pick up the CO2, they make those macromolecules out of carbon, and then we eat those macromolecules, and it's a nice carbon cycle, we call it, because the carbon just cycles back and forth through. You probably heard about that somewhere along the way. So, check out this reaction. I'm pretty sure this is not the reaction that you learned about. How many of you actually had a botany class? If you're a biology major by now, you probably already had a botany class. So is that the reaction that they showed you in botany? Not exactly.
What does X stand for?
Beautiful question. In botany, what did X stand for? This chemical you know, right? Oxygen. I mean water, sorry. Put oxygen in there in place of the X. If you do the same thing over here, you release 12 oxygens. Usually we're going to write it like this. Right?
But they used oxygen for at the end there with 6H2O.
Okay, so you flip these guys around. So because out of photosynthesis you get some water production, you get some oxygen production, and then you make a macromolecule out of it, the way that you learned about photosynthesis. The macromolecules are what we eat, these guys. The oxygen is what we breathe, these guys, and the natural process of photosynthesis releases a little bit of metabolic water as it does that reaction. So you learned about that, but in this class we have to use X. Because what you're going to learn in this class is not all photosynthetic organisms do photosynthesis the way that you learned about it in botany. And the real big question is, what is this chemical? Because whatever this chemical is determines what this is. The way you learned about it in botany, this was water. And they might have just said 6 waters feeds in and they'd leave the water off on this side, but that process actually generates some water as it's going through. So if you want to build a balanced equation, this is the way it should look. But we already talked about some organisms that use something that looks like this. Some of those guys are also photosynthetic. If they do photosynthesis, what do you think this is? Yes, we just looked at that equation just a few minutes ago, a little bit the same. And so basically they're doing the same thing. So now I have to give you a new definition of photosynthesis. You have to scratch number 3 off of that list, because not every organism that does photosynthesis actually releases oxygen. Those that do that reaction that we just talked about, that make hydrogen sulfide, or use hydrogen sulfide, they don't make oxygen in the process of doing photosynthesis. Yeah.
But I'm not. How am I going to ask this? The product though, whatever they happen to be using, it comes out.
You're talking about this as the substrate or this as the product?
The product is like 12 and then insert element there, but that's a pure element, always.
So we did this, hydrogen sulfide and sulfur would come out. Typically that's the way it works. You know, I don't like to say always because there's so much diversity. Somebody's going to do it different sometime along the way. But typically that's the way it works. Okay. The X might be bigger. If the X is bigger over here on this side, meaning that there's more than just one element there, then the X is going to be bigger over on this side too. Okay. All right, so a couple things that we need to think about when we're thinking about photosynthesis. We can break it down into two stages. Photosynthesis has two different parts. And one part requires light. You know photosynthesis requires light, but only one of the parts actually requires light. And those are the chemical reactions that are called the light-dependent reactions. For those to happen, there has to be light. But there's another set of reactions that don't require light. They require light to have been around at some point to charge that side, and they get energy from that side, but it doesn't have to have light while those reactions are working. So many of you think plants are photosynthetic. They do their photosynthesis during the daytime, but in reality they do part of it during the daytime. If you go back to your botany notes, you'll probably see that, and it'll make sense now. They do part of the photosynthesis during the daytime, and then when the sun sets, they do another part of the photosynthesis, and it's because of the biology of the plant that you learned about. Sometimes they need to open the stoma, and they need to take different things in, and so they'll harvest the energy from the light when the light's available, but if they open the stoma during the daytime when it's hot, the plant will actually dehydrate, and it'll likely die unless it's got some special systems to keep it from doing that. So it harvests the energy, it holds the energy, and then when it can open the stoma, and if we're talking about plants, it's going to open the stoma, it's going to take in some CO2, and it's going to do the rest of the photosynthetic process that you know about. And so those things that happen in the dark are called the dark reactions. They don't need light to be carried out. None of the enzymes that do those reactions need light, but they need products that are harvested, the energy that's harvested from the light when there is light. And so we've got two parts, the light-dependent reactions and the dark reactions. The light-independent reactions, those dark reactions, use the ATP that's synthesized during this process up here. Okay? So the light-dependent reactions make ATP. They harvest energy from photons, they make ATP, and then the ATP can be used to energize the systems that do these dark reactions. So you build one product, you build several, but one of them is ATP. And that ATP isn't used during the daylight. It can be by some plants if they've got systems that can deal with it and do it. But it can also be done in the dark. Those reactions will continue. If you put the plant under a light, they'll do the light reactions. They might be doing some dark reactions or light-independent reactions. But if you put them in the dark, those light reactions will immediately stop. The light-dependent reactions will stop, and at that point only the dark reactions can occur. So the light-independent reactions, or the dark reactions, are when carbon fixation happens. And I just described the process in plants because you're more familiar with plants. It's not really too much related to biology, but it happens because of the biology of the plants. And so similar things happen in algae and other photosynthetic organisms. They're typically, not all of them, and this one I can say definitely not all of them, do it exactly like this. And we'll look at some different ways that they do it in just a little bit. But some of them do it a lot like plants. So that's what's beneficial about this table, is that it kind of helps you compare something that you know. You're pretty familiar from other classes about plants and algae. If nothing else, we eat them all, or something we eat ate them. So you're familiar with them in some way. You know about the structure of plants. Down inside of plant cells, what is that structure that harvests the energy from light? There's a chemical down inside that's called chlorophyll, and where's the chlorophyll stored in the plants? In the chloroplasts. So chloroplasts are little organelles, kind of similar to mitochondria, except for their function is to absorb light from energy. They've got lots of little membranous structures down inside. On the outside, they actually have two lipid bilayers, two membranes on the outside, and down inside they've got little compartments that are enclosed by membranes that are called the thylakoids. And down inside of the thylakoids, They're analogous to the cristae in mitochondria. They're there to give extra surface area so extra proteins can be embedded in those surfaces so you can get proton pumps doing what proton pumps do and pumping protons across membranes. So the thylakoids are there, they're down inside, and outside of them is two membranes that house those. That's way back in chapter 3. If you want to look at the structure of those guys again, go back and look at it. And so when plants do photosynthesis, they actually use two systems that we just described in different terms. They use photosystem 1 and they use photosystem 2, and we'll look at those specifically in just a second. The primary pigment that harvests the energy from the photons in plants is called chlorophyll A, and they use water as a source of electrons. So water is the electron donor in this case. Water has the reducing power, and so that's what plants use for their reducing power, the source of those electrons. And most plants fix CO2 by a process that's called the Calvin cycle. We'll actually look at it here in just a little bit if you had never heard of that before. And then plants also have some accessory pigments, and those are called carotenoids. So you've eaten a carrot. Carrots are what color? Orange. And the carotenoids tend to have a kind of orangish pigment as well. It's said, I haven't tried it, you guys can if you want to, but if you eat excessive amounts of carrots for a long period of time, your skin will actually start to pick up an orangish sort of pigment. And it's those carotenoids from the plant cells that are actually being absorbed and presented out there. And I don't think you probably become photosynthetic at that point, but let me know if you feel like you have more energy once you do that.
I have an autistic cousin, and he stopped eating anything that wasn't orange, so he sweet potatoes and carrots, and he actually started turning orange.
I could live off sweet potatoes. I'll tell you another story about China another day. But and then, so those are the guys you're familiar with. They do stuff that you already know. And then there's somebody that you're kind of familiar with because they're a lot alike. They're the cyanobacteria. You looked at those guys in the laboratory, way back, third, fourth lab in the lab. You looked at a number of cyanobacteria, and they do photosynthesis too. And you saw some of the structures that they use for photosynthesis. I had to lie to you just a little bit at that point. Remember, I'm sorry, but you wouldn't have understood if I told you the truth at that point. Remember the definition of bacteria. What's the definition of bacteria? Prokaryotes, single cell, one more thing. No membrane-bound organelles, except for the cyanobacteria. If you look down inside of the cyanobacteria, you start seeing some membranous structures, some compartments down inside that have membranes surrounding them that start to look a little bit like thylakoid stacks in a chloroplast. And I couldn't explain it to you back then because it would have blown the definition of a bacteria out of the water. And that's why, again, I don't like to say, yes, that's always the case. There's somebody that breaks the rule. For no membrane-bound organelles, it's the cyanobacteria. They are prokaryotic, and they're single-celled. There's one other part of that definition you didn't give me, too. They have peptidoglycan in their cell wall, by definition, bacteria do. And they don't have membrane-bound organelles. And these guys have some things. We don't like to call them organelles, but they look a lot like organelles, and they're compartments that are surrounded by membranes. So it's really stretching the definition to not call them a membrane-bound organelles. And I didn't want to tweak your brain too much at that point, so I didn't tell you about them then. But now you know there's another exception to a rule that we've established as humans. Okay, the cyanobacteria are familiar too because they do photosynthesis pretty much analogous to the plants that you know about. And so it's one of the reasons that we looked at them close together at the same time. And they also use chlorophyll A as their primary light harvesting pigment. And they do this process, I skipped it over last time, but both plants and cyanobacteria do it. They do a non-cyclic photophosphorylation. You got that term photophosphorylation last time we were together. It's a process that makes what? No, it uses phosphorus to do something else, to make something else that you know about. ATP. It uses phosphorus to recharge ADP to make ATP. Good. And so they do that process, non-cyclic photophosphorylation. When you hear that, that means making ATP. And these guys can do that in both of the photosystems. In other words, photosystem 1 makes ATP, and photosystem 2 can also make ATP. Okay? And they use the Calvin cycle. Again, that's kind of just two words. You may not know what it is unless you were one of those that raised your hand in botany. I'm pretty sure you learned about it there. We'll learn about it in just a second. They also have carotenoids as accessory pigments, and they have some others that are called phycobilins, a group of different proteins that can take energy. That basically pigments all absorb light. These guys can use different photons of light and harvest or help harvest some of the energy from those photons. So these guys, I said you're familiar with, the plants, algae, and the cyanobacteria, right? Because they do photosynthesis the way that you know it. Look what we call the type of photosynthesis they do. We call it oxygenic photosynthesis. What does oxygenic mean? Photosynthesis that does what? I heard two different answers. I'm going to wait for you to get me the right one. Uses oxygen? Produces oxygen. Okay, good. I thought I heard some say use. It doesn't use it. Genic means to generate. It's the root for that. So these guys, when they do photosynthesis, they produce or generate oxygen. All right, and that's the kind that you know of. And so the other kind are these guys that do anoxygenic photosynthesis. What would that be? Photosynthesis that does not produce oxygen. And I don't want to go back there, but if you look at that equation, that's why we have to put the X in there. Some organisms do photosynthesis, but they don't produce oxygen. And now you know what different things that can feed into that photosynthetic process rather than water feeding in as the electron donor into those reactions. Right? Talked about one of them. We'll look at some other things here. So now we're going to talk about these guys, and this table really has 5 columns. The first one's over here, and then we just looked at the second column, but you could break the second column into 2, and we could break this into 2 columns as well, and look at them together. And so the anoxygenic bacteria include 2 groups of bacteria. One group is called the purple bacteria, and the other group is called the green bacteria. And they do things a little bit differently. And they do things differently because of the pigments they have, but they also do things, we break them into those groups and call them those colors because of the pigments that they have. So if we just look at the purple bacteria first, these guys are anoxygenic photosynthetic organisms. And again, these are bacteria, but look close. Down inside the cytoplasmic membrane, they've got invaginations down inside of the cytoplasmic membrane that are designed to hold lots of chemicals that can absorb photons. So again, we don't like to call them organelles, but think down inside of the mitochondria. What did you see down inside of the mitochondria? Cristae that have lots of invaginations of that inner membrane. These guys are also double membraned organisms, and they've got those same kind of invaginations that are loaded with the enzymes that are able to harvest photons of light. And so again, we're getting a little bit close to the edge of bacterial definition here, but it basically, in all those cases, it increases surface area so you can get more enzymes, more chemicals, more substrate to be able to do the reactions that these cells are designed to do. So when we look at the purple bacteria and the kind of photosynthesis that they are able to do, they only do, or typically only do, photosystem 2. If we look at the plants, algae, and cyanobacteria, they do both, photosystem 1 and photosystem 2. Again, right now, that's just words, may not mean anything unless you've taken a botany class somewhere down the road. But we'll look at it in just a second. The light-absorbing pigments that these purple bacteria have are called bacteria chlorophylls. Not chlorophyll, but they're molecules that look like chlorophyll that are made by bacteria, so we call them bacteria chlorophylls. And they do something interesting. Over here we saw photophosphorylation, non-cyclic photophosphorylation. These guys, to generate reducing power, run the electron transport chain backward. How the heck does that work? Got enzymes that can do the reverse reactions of the electron transport chain. What would be the product of the electron transport chain if you were to run it backward? NADH and FADH2. You'd be reducing the oxidized forms of those chemicals, and those reduced forms of those chemicals that feed into the electron transport chain again have reducing power. They're electron carriers, and they can carry those electrons off to some other chemical reaction. So that's the way the purple bacteria do it. They run the electron transport chain backward. That means they have to have a whole different set of enzymes that can do that. But ultimately, they can make NADH, and they can make FADH2 by running the electron chain backward. Beautiful. All right, and so when they do that, they're going to get energy from a number of different places, and this depends on which organism. Some of them use that hydrogen sulfide. Some of them use elemental hydrogen gas, and many of them can use other organic compounds along the way. To fix the carbon, because that's part of the definition of photosynthesis, these guys also use the Calvin cycle. So fixing CO2 in these guys works just like it does in the plants, the algae, and the cyanobacteria. They also have the carotenoids as their accessory pigments. All right, one more group to look at. Don't lose me yet. The other one is the green bacteria. These guys have a cytoplasmic membrane, and on the inner surface of the membrane are accessory pigments, usually not quite as much invagination down inside of these cells. They run the photosynthesis, and their system looks like photosystem 1. So both of these, the purple bacteria and the green bacteria, do half of the photosynthetic process that plants, algae, and cyanobacteria do. And they each do a different half of it, and they each are able to get the energy that they need out of that. They too have the bacteria's chlorophyll, and these guys go back and they use a non-cyclic photosystem to generate that reducing power. We'll look at it in just a second. And to fix CO2, they do something different. Remember, in your cells, in your mitochondria, what happens when the pyruvate comes into your mitochondria? Pyruvate's got 3 carbons, and by the time it goes around the circle, how many of those 3 carbons are left? If it goes around enough times, none of the carbons are left. Why not? Because CO2 is kicked out. 3 different places. You lose a CO2 at the intermediate step when you're taking pyruvate down to the acetyl group that you guys just looked at in that homework assignment. A couple steps down on the TCA cycle, you kick out another CO2. A couple steps down, you kick out another CO2. You fed 3 carbons in, you just kicked out 3. And after a couple cycles, all of those carbons that came in with pyruvate are going to be kicked back out of that cycle. So if you ran that TCA cycle backward, what would happen? If you reverse the TCA cycle, what would happen? You'd fix CO2 at each one of those places that you, in the normal TCA cycle, kick CO2 out. So some bacteria can run the TCA cycle backward. Again, it means they have to have enzymes that can catalyze those reactions in the opposite direction. They can do that, and as they do that, basically they build pyruvate. They fix 3 carbons, and every time they do that, they push out an acetyl group, and then it can be converted to pyruvate, and it can go on up the process of either lipid metabolism or gluconeogenesis, which can then make amino acids. So lots of diversity out there that you may not have heard about before, and hopefully you're getting an idea about that now. So photosynthesis, you already know, is involved in capturing the radiant energy. Capturing the radiant energy from light. Usually we say sunlight, but this light can work too, depending on the organism. And basically each plant is going to be looking at different wavelengths, and it depends on what those pigments are, which lights, the wavelengths they prefer. Some use chlorophyll, plants, algae, cyanobacteria. The bacteria use those bacteria chlorophyll. They absorb wavelengths, different wavelengths than chlorophyll. And then the accessory pigments may absorb even additional wavelengths, like the carotenoid, and others tend to absorb different wavelengths. And the phycobilins are going to be in those red algae that you looked at in class, and the cyanobacteria, again absorbing different pigments of light. Some of them also use antennapigments. They're a reaction center structure. Their function is to grab the energy in the photons and funnel it into the rest of the photosynthetic pathway. We're actually going to look at how that works here. So I know right now, if you haven't had a botany class, or if you haven't taken 8th grade biology in recent years, you probably forgot a lot of this. And so we're going to look at how this works, and just to make sure that you've got it. So we're talking about photosynthesis, and if you look back at the previous figure, that table, it talks about photosystem 1, photosystem 2, and inside of those photosystems are reaction center pigments. Pigments that are down inside it, and what these are is areas inside of the cell where these pigments have come together to do their job. And the guys donate electrons into the electron transport chain. They're actually producing excited electrons, charging up electrons. Remember how we talked about flow of electrons, high energy electrons, low energy electrons? These pigments are harvesting photons, the energy from photons, and transferring that energy to electrons to make them high energy electrons. And those high energy electrons then can go and drive the electron transport chain. You already knew that because that's basically what's happening in the mitochondria that you've learned about in AMP. High energy electrons are being brought over to the electron transport chain to run it so the proton pumps can work and the rest of the process can work. And the pigments that play the predominant roles in that are chlorophyll A and the bacteria chlorophylls. The cyanobacteria have photosystems in the membrane, the stacked structures that are similar to those that are called thylakoids in plants. And if you look down inside of the chloroplast, in the center of the chloroplast, that open area, you see those thylakoid stacks. So, remember early on, chapter 3, I think it was, we started talking about this theory called the endosymbiont theory. And the endosymbiont theory says that mitochondria, chloroplasts, and a few other organelles, once upon a time, were small prokaryotic organisms. And they couldn't harvest materials from the environment very efficiently. They could do it enough, obviously, to be able to survive. But they couldn't do it very efficiently. But what they could do is when they got those materials, they could harvest a lot of energy from those materials. And so these small prokaryotic organisms moved into a bigger cell. And that bigger cell was very efficient at harvesting chemicals from the environment. It could make the enzymes that could break down all of those macromolecules, the cellulose, the amylase, the proteins, all of those macromolecules that we just talked about. The big cell had the enzymes to do that, and the big cell could bring those monomers down inside, and it could squeak out a little bit of ATP, enough to get by, 2 ATP per glycolysis reaction, from glucose to pyruvate. And it made enough to get by, but now this little cell that could use the pyruvate moved in. And now there was lots of pyruvate available to that little cell, and when the little cell got it, it could make tons of ATP, 36 ATP out of one of those pyruvates. Okay, half of that out of one of those pyruvates. And so it could make a lot of ATP, and the little cell said, hey, this is good, somebody's feeding me, I'm going to play nice, I'm going to share some of my product with the big cell. And the little cell shared some of its ATP with the big cell, and so they developed this relationship. And it was good for the little cell because now somebody was feeding it. The little cell could make lots of product. It could feed it back to the big cell. And therefore, the theory says this symbiotic relationship resulted, and that's how we got mitochondria. Same thing for the chloroplasts. The chloroplasts were efficient at harvesting photons of light. The big cells obviously couldn't do that. The chloroplasts figured out, hey, if I go inside, somebody's going to feed me organic molecules. And then the chloroplasts shared some... the big cell's going to feed it CO2. The chloroplast then shared the ATP that it made, the carbohydrates that it made, and so again, that's what the background is for this endosymbiont theory. I know for a fact I wasn't there if this did happen, so in my mind it's still a theory. You grab onto it and use it however you want to, but I do want you to know about it. Most of you are going into the medical field, and a lot of doctors, a lot of nurses are actually Christian. Some of you I'm sure are, some of you may not be. But when you get into the medical field, if you go to a high-level hospital to work, you're going to be challenged in your faith. And this is one of the theories that they're going to use to try to persuade you. So you need to think about it and think about some other ways that what the theory says happened could have happened, and figure out what the evidence there is to support. There are people that argue both ways. I've got friends who argue both. I just want you to know when you get out there in the real world and you're not buffered by a UMHB environment, you're going to hear people saying that evolution has to happen. We can see it in those organisms, those cells that have mitochondria. We can see it in those cells that have chloroplasts. They're going to argue that point of view. Want you to know that. So when we come back on, do you guys want to have class on Saturday? I really feel like we need to have 3 classes every week. Would you be willing? No. All right. I'll see you Tuesday then.